Hello, I'm Fraser Palamara, and I'm joined today by Island Pharmaceuticals CEO and Managing Director, Dr. David Foster. David, could you give listeners an overview of Island's Phase 2AB PROTECT trial in dengue fever?
Yeah, I'm more than happy to, Fraser. Thank you very much for the chance to be here and give you guys an update. So, Island Pharmaceuticals is developing a lead molecule called ISLA 101, and we're developing this molecule to see if it could be either a prevention or a treatment for the dengue fever. There's about 400 million dengue infections a year. About 100 million of those require some form of intervention, and there's no intervention. There's no small molecule that you could take that would be an effective antiviral. Vaccines have been problematic being developed. They're very challenging. So we think there's a real opportunity and a real need for a small molecule. The study that we're running is called a challenge study, and we're running this in collaboration with the US Army. We're running this at a site in New York, in upstate New York, in Syracuse. And the way a challenge study works is you enroll healthy individuals, and then you infect them with an attenuated or a weakened strain of the dengue virus. This is a virus that was actually developed by the US Army. And you do this all in a very controlled hospital setting. And one of the beautiful things about this is because you're holding the virus, you have the virus, you can control when people get infected. That also means you can control when they get the drug. So you could imagine a situation where you might pre-treat individuals with drug and then infect them and see if you could block that infection. Or conversely, you first infect them and then you give them the drug and see if this can be a treatment. And we're actually doing both in our clinical trial, the PROTECT study. The first cohort was a prophylactic study where we pre-treated individuals and then infected them to see if we could block that infection. Importantly, at the conclusion of that study, which we ran last year, the FDA required that we have a safety review committee look at the data and see if the drug was effective, if there was evidence of antiviral activity. And if so, then we could move to the second cohort. We did that. We're in the second cohort right now, which is a treatment study. So in this study, we first infect the individuals, and then a week later, we put them on drug. So we are wrapping that study up. We have concluded that study. We're really analyzing the data right now. So it's a really exciting time for us. We are very excited to see if ISLA 101 could be either a prevention or a treatment or potentially even both.
So, David, following the Phase 2A cohort, the Trial Safety Review Council had some interesting findings. Can you give a bit more context to this and what was involved from a trial protocol perspective?
Yeah, absolutely. It's a really good question. So, as I mentioned, we did a prophylactic cohort with 4 individuals, which would lead to a therapeutic cohort in 10 individuals. Because we're infecting people with this weakened strain of the virus, we always want to make sure that people are safe, right? So the FDA said, we'll let you do your first cohort with 4 individuals, but before you start that second cohort, we want a small group of dengue experts to take an interim look at the data, look to see if it was safe, look to see if you achieved the appropriate blood concentration, and then third, to see if you saw evidence of antiviral activity. And only if they answered yes to all 3 of those questions would we get to move to the treatment cohort. So I've given you the punchline already because you know we're in the treatment cohort or we're concluding it. But importantly, the Safety Review Committee did see evidence of antiviral activity that satisfied the FDA, that enabled us to move into the treatment cohort. So very exciting times.
So lastly, David, the company is awaiting those final results, which are anticipated shortly. What is the process to obtain these?
Right. So we began dosing this cohort earlier this year. We infected the individuals. We dosed them. We've concluded all of that. We've been taking blood samples all along so that we could do the appropriate analyses. And there's a variety of analyses that are going on right now. The important ones that we're very interested in are the pharmacokinetic data. These are the data that tell you what's the blood concentration of ISLA 101 in these individuals. The second part that's very important is what's the virus level in these individuals. The pharmacokinetic data has actually come in, and I'm very happy to say that the dosage regimen that we used in the study was very effective. We nailed the dose. We hit the target dose and target blood concentration that we were hoping to achieve. So that's very important. As to the virus level, we're still waiting on those data to come in. They're starting to come in. It's going to take another week or so before we get all of that. Then what happens when we get the data in our hands, we will review it, we will audit it, we will make sure that data is pristine, that there's no mistakes. because then it's ultimately going to go into my clinical trial database. Once it's there and we're happy with it, we lock that database. We can't make any additional changes to it, so it's fixed, and then we get to unblind the study. So in a matter of weeks, before the end of May, we will be unlocking the database. We'll look at the entire study from the prophylactic all the way through the treatment cohort. Both cohorts will have the unblinded data. We'll know who's on drug, we'll know who the placebos are, and we'll know how they responded to the viral infection. So very exciting times. I know I've said that a few times, but it really is. I mean, we're just weeks away from having fully unblinded data out of this study.
David, it's been a pleasure as always to chat today. Thank you for joining us.
Thank you very much. It was my pleasure.